Okay, so now Amanda is going to present her work about investigating neutralizing antibody to SARS-2 in domesticated and wild animals. So we're excited to listen and really looking forward to it.
Thank you very much. Thank you for the introduction. So we've already had really nice introduction to SARS-CoV-2 in animals and why we're interested in looking, particularly because we know that SARS-CoV-2 originated in wildlife, perhaps through an intermediate host before jumping into humans. And since 2020, there's been a large number of cases of SARS-CoV-2 in animals, zoonotic transmission and also reverse zoonoses. And at APHA, we've been looking at SARS-CoV-2 cases in animals as well, and have been describing some of those cases. And of course, the most importance is the potential for evolution of new variants in these new species, and the potential for emergence of persistent reservoirs, and then the spill back to humans. And so the aim of this work that we've been doing is to look at some assays that are specific and sensitive enough that can study prior exposure and looking for SARS-CoV-2 in domestic and wildlife species, and look at the prevalence in non-human populations. And of course we know that coronaviruses in general are a highly diverse family of viruses with a wide host range, and this presents some challenges when we're looking for SARS-CoV-2 serology in animal species. And we know that they result in respiratory and enteric diseases, and there's obviously the potential for cross-reactivity of antibodies to SARS-CoV-2 and other coronaviruses, and they're not all known for all the species. And so we were looking at some serological assays to see which ones would be good to use for animal serology. And at APHA, in collaboration with others, we did a study looking at human sera, and this was early on in the pandemic, and looking at a virus neutralization test with the early virus, and also looking at the lentiviral-based pseudotype viral neutralization assays, as well as a commercial ELISA and an in-house whole antigen ELISA. And the data from the human serology side showed good comparison between the virus neutralization test and the pseudotype virus neutralization test, and also correlated well with some of the ELISA, but showed that really when you combine the tests together, you get a higher detection rate. And so we wanted to take forward the virus neutralization test for screening for animal serology. However, we also, later on, as the years went on, a SARS-CoV-2 antibody ELISA, double antigen multi-species ELISA for animal sera became available. And so we wanted, in an ideal situation where we could screen using this ELISA and then confirm with the virus neutralization test. And I'll explain with the data that we've got how that has performed. And so we collated samples from a range of sources. As the National Reference Laboratory for SARS-CoV-2 in animals, we had some cases, and we were able to use samples from there. And also running the PET travel scheme for rabies, we have availability of sera from multiple countries. And we also obtained some samples from the RVC and BioBest for some specificity testing. We were also able, through research and surveillance projects, to obtain wildlife samples through wildlife management activities and engaging with several wildlife managers and hunters, gamekeepers and wildlife trusts, and look at priority species such as mustelids, canids, cervids. And we also had a collaboration to collect samples from fruit bats in Nigeria, and these were pre-pandemic sera, and this was originally for looking at sera prevalence of Lagos bat lyssaviruses in e-health and fruit bats, and I'll show some of the data that we obtained from that. So firstly, looking at some specificity testing. So we started off in the domestic animals looking at cats. So from the pre-pandemic a collection of samples that we had, we did find that there was one that was positive in a domestic cat from Hong Kong, and this gave a neutralisation titer. And when we look back at the collection date of this sample, it was from November 2019, and so that was quite interesting to see. And other studies have shown that that actually, when you date back when the initial incursions were of SARS-CoV-2, it falls within that time period. We'd also tested the sample on a immunocomb for feline coronavirus, and it was also positive. So it's possible that either this test was not specific enough and it was picking up SARS-CoV-2, or this cat had both was infected with both at some point in its life. We then also took sera from cats early on in the pandemic, January to June, and they were negative. And then we had some feline coronavirus positive cats that we obtained from BioBest to look at the specificity, and showed that the SARS-CoV-2 VNT didn't pick those up, which was good. And so there was no cross-reactivity there with the feline coronavirus. When we looked at the similar sort of sets of samples in dogs pre-pandemic and early pandemic, they were also negative for SARS-CoV-2 on the VNT, and the canine coronavirus positive dogs that we obtained via RVC were also negative on the SARS-CoV-2 VNT. So apart from that one Hong Kong cat, where we're assuming that it is possibly did have SARS-CoV-2, it showed good specificity. However, we also obtained this wildlife sample from fruit bats from Nigeria, collaborating with the university, and we found a proportion had VNT titers positive for SARS-CoV-2 in a similar titer to what we saw in that cat from Hong Kong, as an example. However, these were all pre-pandemic sera. We were then able to subsequently collect fecal droppings from the roost and detected a lineage D Betacoronavirus from this collection, and so there's a possibility that we assume that is what we are seeing in the VNT for SARS-CoV-2 is a cross-reactivity with the lineage D. So moving on to some of the cases of domestic animals. So the first one was a cat that was positive for SARS-CoV-2 and was published by Glasgow University. And we also collaborated to share some of the sera and confirm the results, and found that confirmed that VNT was positive for SARS-CoV-2, confirming that the infection, and in this case, both the cat and the owner made a full recovery. So just showing an example of the VNT giving a positive result in a positive sample that we believe. So then looking at the companion animals, so we were able to access sera from 3 different years. So we looked at the peak in the UK, peaks of UK human infections, and then we collected sera from around those times each year, and we had a proportion testing. So we then had the ELISA at this point in time, and so we wanted to test the sera by screening, all the sera by ELISA, and any positives we then took forward for the VNT, but we were also interested in the ones that were negative. And what we could see is for cats here we had about 8% positive on the ELISA, and we took a proportion of those positives, and less than half of those were positive by VNT as well. However, some of the ELISA negatives were also positive by VNT. So in the graph on the right, we can see at the bottom there was a large proportion that were ELISA negative and VNT negative, a small number that were ELISA positive and VNT positive, but there were some that were ELISA negative and VNT positive, and vice versa, ELISA positive and VNT negative. So this really just trying to highlight that both tests really don't always match up, and a similar pattern, in fact, a worse correlation in the dogs that we saw here. We've had nice introductions to feline coronavirus already, but I just wanted to highlight on this slide here that we had we also screened the sera using the feline coronavirus immunochrome and found a large proportion were positive for feline coronavirus. And we did submit some of these samples to Lucy and Marsha for analysis that was mentioned earlier today. And so out of the 10 SARS-CoV-2 ELISA positives, 9 of them were also positive on the feline coronavirus test. And when we looked at the ELISA positives and feline coronavirus positives and tested those by VNT, we saw that they were positive as well. So moving into wildlife, we collected several samples from deer species. So originally we were looking at swabs and tissues from deer and wanting to screen for SARS-CoV-2 and other coronaviruses using a pan-coronavirus PCR, but we also had the serological samples to test with the ELISA and the VNT. And in this sample set, we used the ELISA to screen and then the VNT to confirm. So we only tested some of the ELISA positives by VNT here. And we found that not always did we get positive VNTs when we had positive ELISA results, but there were 10 that were positive by both. And when we look at those, we tested them by Delta and Omicron. And the ones boxed in red are the ones that gave much higher titers. And so, but what we wanted to look at was to see whether when we use the whole virus of Delta and Omicron in the VNT, can we determine which deer or which variant the deer was infected with? And we don't really see a pattern of one being more infected with a higher titer than the other. This just shows where in Great Britain we collected samples from and where the positives were found. And of course, there's a lot of concern about white-tailed deer in the US, but we haven't found any SARS-CoV-2 in deer species in the UK. Some of the ACE2 receptor mapping has shown that they are susceptible to SARS-CoV-2 infection, but we don't know whether they have or not. We haven't seen any reports yet. However, we're also interested to know what other coronaviruses deer species have. And there was one report led by APHA, and in a fallow deer, a Betacoronavirus was detected, and bovine-like Betacoronavirus. And so it's possible that what we're seeing in the serology is a cross-reactivity, but without further information, we still cannot be sure. So some of the other wildlife that we tested in multiple species, we also found some more seropositives. And looking specifically at the badgers, we found some ELISA positives in the badgers, and however, these were not VNT positive, but we did find a VNT positive that was not ELISA positive, and we collaborated with Margaret and Grace to look at these badger sera samples and look at the pseudotype VNT and multiple variants, and they were all negative. And so we're also interested to see what other coronaviruses these badgers might have. And so I'm very interested to look at the poster on what coronaviruses might be in badgers. So in the sample set that we had, we only had throat and nasal swabs. So there could potentially be a sampling bias there if the badgers are potentially infected with an enteric disease of SARS-CoV-2. And some of the final data sets I wanted to share was when we experimentally infected ferrets with SARS-CoV-2. So this is with the Delta variant, and there are two different experiments on the left and the right. On the left, two groups of ferrets infected or exposed to SARS-CoV-2 contaminated skin. And really what I wanted to highlight here is the bit circled in red. So The black bars represent VNT and the grey bars are ELISA. And what we see is that there's one ferret in each group that was positive on the VNT but negative on the ELISA. And so we do see that pattern. And in further experimentally infected work with Delta and Omicron, we saw that in this experiment we directly intranasally inoculated ferrets, and then after 5 months we re-inoculated them. And we took serum samples over several time points throughout that duration. And the ferrets were all positive prior to reinfection. And after 2 weeks and 4 weeks, we saw that the VNT positive from the directly inoculated in contacts, but the ELISAs were only positive on the direct inoculated and not in the contacts. And so that was with Delta, and with Omicron, we saw a similar thing, but the ELISA was only positive in one of the directly inoculated. As part of this big experiment, we also did a heterologous reinfection. So we directly inoculated with Delta, and after 5 months, we reinfected with Omicron, and vice versa. And what we have here is with the VNT positives for both variants in the directly inoculated contacts, and ELISA positives only in the directly inoculated not the contact. So a similar pattern. What is interesting is on the right-hand side, we have one contact that is positive on the ELISA as well, and that is the only one contact that actually got infected in the experiment. So you can see there it wasn't protected. The final piece of data I wanted to share today was a collaborative piece of work under the COVRIN One Health EJP project, where a panel of sera was prepared and delivered to 5 different laboratories and tested. And we tested this in exactly the same way as we've been doing all our surveillance work. So screening with the ELISA and then following up with the VNT. And we saw here that again, a similar pattern. We saw some ELISA positives, but VNT negatives, and some vice versa. And some of these were from pre-pandemic field sera, and some were with experimentally inoculated Betacoronaviruses of not SARS-CoV-2. And so we were detecting, but not of. So if we combine the results from both tests, we don't get anything incorrect in this panel. And so just to conclude that the ELISA and the VNT results here don't always corroborate, and we can't rely on only one test. And so positivity in either test may warrant us wanting to try an additional test using another assay. And so the pseudotype VNT may be more specific for SARS-CoV-2, which we saw with the evidence we've got from the Badger sera, and there is scope to test available sera using these systems going forward to see what that looks like. Another thing to consider here is determining the cutoff for positivity, and this is important when we want to interpret the data and say at what threshold are we going to call this positive, and this may be different in different species, and particularly when we're looking at exposure of different other species to other coronaviruses and the cross-reactivity that we're seeing. And so just overall, the take-home is really that the potential for cross-reactivity in these assays could be due to antibodies that are produced against animal exposure to uncharacterized coronaviruses. And this may be difficult to interpret as a positive or not. And yeah, we'd like to continue this animal surveillance for SARS-CoV-2, particularly in light of any future.
So thank you very much for all this big work that you have done, that you showed us. Oh, there's plenty of questions. So.
I know, lovely presentation. Really interested in your Nigerian bat serology. So 20% positive by neutralization, I think, from my mental maths. The sequence that you found, was it such high homology to SARS-CoV-2 spike to explain the neutralization?
So there wasn't a huge high homology when we look at spikes. So the phylogenetic tree that I showed you was just on our DRP, but we did we did attempt sequencing the whole genome, and we didn't manage to get the full spike sequence out, but we did get a large portion of it. It wasn't that similar, but we did identify some areas of homology in some of the domains.
So my suggestion is that is not the explanation, and there is another virus inhabiting those bats that obviously has higher homology to SARS-CoV-2.
It's potential.
Yeah. So keep looking.
Yeah.
Sorry, we ran a bit over, so we need to stop the question. I'm so sorry, but I'm sure Amanda can answer later. Sorry for the disappointment, guys. We need to move on. Sorry, thank you, sorry, thank you. So is this session? Yeah, okay. So our session of Betacoronavirus is over now, so we are going to move forward to the flash talks.